Okay.
Okay, thank you. So, thank you for inviting me. And I'd like to talk about the proliferation imaging and the genetics. And as already Yuval explained very nicely, but important thing in the corticogenesis, the first phase of cortical neurogenesis is the neuronal proliferation and also apoptosis. And most of neural cells in adults are produced in the first and the early second trimesters. And for adjusting the cell number, physiological apoptosis occur. Now, the most different point in the cortical neurogenesis between mouse and human is the outer subventricular zone. Now, the outer subventricular zone in humans is wider and wider during between 11 and 17 weeks of gestation. Now, there are two stages of human cerebral cortex development during neurogenesis, and first stage is like a mouse, and this is called as continuous scaffold stage. And in this stage, is a completely the same as mouse brain, and the conventionally this is believed for a long time as a cortical migrational phase. However, in humans, next stage appears this second stage here, which is specific in the primates, and this is called as discontinuous scaffold stage. And in this stage, there are many radial glia in OSVZ, here OSVZ, and the radial glia, they jump up and divide themselves and produce many neurons by cloning, which are going towards upper cortical layers. Now, however, here you can see this is the FGFR gene variant, thanatophoric dysplasia, at 17 weeks of gestation, and you can see the abnormal temporal lobe focal overgrowth is seen, like here. But you know, the earlier than this, 13 weeks and 3 days, suspect of the thanatophoric dysplasia, and here this is the brain, and you can see the many lines here, the very abnormal lines, and the overgrowth occurred before 16.5 weeks of gestation. So I think, you know, normally the before 16 weeks of gestation, only deep cortical layers can be created. However, most probably migration stage was accelerated by FGFR3. Now, you've already told about the PI3K AKT mTOR pathway, and mTOR is a mechanistic target of rapamycin, and the pathway is very important for thinking about the proliferation disorder. And in a nutshell, the mTOR pathway serves as a central regulator of the cell metabolism, cell growth, cell cycle progression, and cell proliferation and survival. And a bunch of recent papers provided evidence that this regulated mTOR cycle resulted in alterations of the motile cilia and asymmetry defects. And normal brain development requires careful control of when brain cells divide and when they stop. So the protein mTOR acts as a growth control switch. When this switch is broken due to the genetic changes, cells multiply too much, the brain develops abnormally, or understanding this mechanism help us recognize and manage these disorders imaging and through genetic testing. And those are genes acting as regulators of the mTOR pathway, like mTOR itself, and the AKT1, AKT3, and PIK3CA, and PTEN. And I would like to share with some germline the variant cases. So germline variant, it means de novo variant. The specifically refers to the germline, the variant that arise spontaneously in the parental gametes. And the de novo variants already, they present from the fertilization and affect every cell in the developing fetus. So the case, this case already, published many years ago, and this is the big head macrocephaly at 18 weeks of gestation. In the 18 to 20, you can see the many of the polygyria and the asymmetrical pathological sulci and irregular shaped ventricle. And this, the amniocentesis showed that mosaic paternal UPD of 10q plus P10 variant, and P10 is on 10q. So I think that this case had the mosaicism of LOH and non-LOH, and if all cells were LOH, the fetus cannot be alive. However, this fetus was alive until termination at 21 weeks of gestation, and this is a diagram of the mechanism of how such an extremely rare fetus with paternal UPD mosaicism of 10q and the P10 variant on the 10q was created. Now, the next case, the megalencephaly, BPD 3.9, standard deviation, and SC 3.3. And here you can see the polygyria here, and also on the Sylvian fissure. And this is an asymmetrical structure, and if you look at the left surface, this is very smooth, and if you compare to the normal brain at 24 weeks of gestation, completely different structure. And 4 weeks later, at 28 weeks of gestation, BPD became over 6 standard deviation, and the cortical developmental disorder was more obvious. And amniocentesis exome sequencing resulted in de novo AKT3 variant in mTOR signaling pathway. So likewise, usually when we have a strong suspicion of the genetic disorder, we can find gene variant from the amniotic cells or the chorionic cells. So the case after birth had thyroid hypogenesis, and we published this the postnatal course as shown here. The next case, this is the BPD 3.8 standard deviation HC 4.7, and here you can see that this is the transabdominal sonography, and there's not so specifically the obvious ventriculomegaly. And here you can see this is the video clip, and if you look at the tomographic ultrasound imaging, and you can see the shallow gyri and sulci are observed, and there's no asymmetrical structure in this case, but this is some suspicion of the genetic variant. And also, furthermore, the corpus callosum was observed to be large and thick. And this is the MRI in the same case. And amniocentesis was done, and chromosome 15, Q11.2, Q13.1 microduplication was found. And here you can see this is the explanation of this partial duplication. And it says that the autism, intellectual disability, epileptic seizures, and schizophrenia, and this is a pathogenic. Usually, the chromosome microarray is positive. We don't go for the exome sequencing. However, I didn't think that those brain findings are due to the 15q microduplication, so we did not hesitate to go for WES. And the result of the WES was de novo mTOR gene variant. So actually, the mTOR is in the mTOR pathway, and here you can see. So this is the double gene variant, the microduplication and the mTOR gene variant. And this is the 35 weeks brain in the same case with an abnormal gyration. Now, the somatic variant mosaicism, already, you've told about the somatic variants, and the somatic variants that occurs post-fertilization. The normal sperm and the normal oocyte, and they fertilized, and after fertilization, some of the cells, they have the genetic variant. And so the amniocentesis, there's no variant. However, somewhat in the brain, the focal brain, the region, there is a mosaic variant confined to the focal region. So now this is a case. The BPD was 2.8, standard deviation HC 3.7. And here you can see I showed this several times, so some of you already remember this case, but the polygyria here and there, and obviously this is very, very the abnormally asymmetrical structure. And moreover, irregular ventricular wall and the hyperechoic the ventricular zone plus the inner subventricular zone. So we did amniocentesis on the same day for the trio basis exome sequencing, and 9 days later, we received the results showing no pathogenic variants, maternal blood and paternal blood and the amniotic fluid, all test negative. And this presented us with a diagnostic puzzle. But here you can see eventually, they terminated the pregnancy, and the histology finding after the autopsy. And we extracted the DNA from the FFP brain tissue, and we did the exome sequencing of the both brain tissues and heart blood, cord blood, placenta, membranes, and the mother's and the father's blood. And we found the mosaicism of the AKT1 variants in both brain, 40% and 43.8%. And actually, this is the histology findings. This is a polymicrogyria, and this is a heterotopia here and there, and subarachnoid neural heterotopia. There's one more cortical layer here. This is a overmigration, and also here inside here around the ventricle, you can see the subventricular heterotopia. Excuse me. So not only proliferation disorder, but the same gene the causes the migration disorder as well. Already, the Yuval explained about that. And the next case, the bioultrasound. Here you can see the hemimegalencephaly and very, very abnormal the asymmetrical structure of the brain, and BPD was 4.3 standard deviation. And here you can see in the coronal image remnant gangrenic eminence and the abnormal the Syrian fissure appearance and abnormal circulation and the irregular ventricular wall. And we've seen, and from all those findings, MCD, especially the mTOR signaling pathway relevant, the MCD was strongly suspected. Now, this is the normal brain, the vessels at 19 weeks of gestation, but in this case, the overgrowth cerebral blood vessels were observed as shown here. So presumably, the overgrowth of the cerebral vessels accompanied the overgrowth of the brain. And by ultrasound, here and there, you can see the overgrowth in some of the part of the body. And we did amniocentesis on the same day, and the same result as the previous the case. However, because of the multiple abnormalities, parents decided to terminate the pregnancy, and we did autopsy as well. And here, from the tissue, the brain tissue, and the other tissues, we found that the PIK3CA gene variant mosaicism. And this is the somatic variant mosaicism, and you can see the right brain 22%, left brain the 48%, and here the buttock tumor 53.8%, but amniotic fluid, cord blood, mother powder 0%. So this is the somatic the variant mosaicism. Now going to the microcephaly, and here you can see this is the case, 24 weeks of gestation. This is very, very old case, and this is the 2013 case. And here you can see this is very, very abnormal at thinking about the 24 weeks of gestation. And this is the case, the 6q partial duplication and 6q partial deletion. And it is quite difficult to find that by G-band the result. And also, the partial deletion case, with microcephaly, this is the 20p 12.3 deletion. And also, you can see the very abnormal the structure with abnormal the findings of the Sylvian fissure, and also this is the asymmetrical structure. And here this is another case, the 4p minus case, and you can see sometimes we can see the microcephaly case from the with the chromosomal partial deletion or duplication, but this case even exome sequencing negative, but this is very, very abnormal microcephaly with migration disorder. Now, of course, the genes like ASPN, gene variant, one of the responsible gene for MCPH, and FOXC2 gene variant. Sometimes, it is associated with microcephaly, and as already Yuval told that the TUBA1A gene variant tubrinopathy. And you remember this, there's a much radial Guyer, and they are jump up and divided themselves and produce many neurons by cloning. However, this is very interesting experimental study of the lissencephaly by using time lapse, and inside OSVZ, and I thought that the rear jumped and divided themselves. However, in lissencephaly brain, the division time is extremely long, and they could not produce proper number of neurons, and therefore neurons in upper cortical layers of the lissencephalic brain are much less than them in the normal brain. And lastly, this is the case at 26 weeks of gestation, the microcephaly, the BPD minus 3.3 and the HC minus 3.8 standard deviation, and very, very abnormal Sylvian fissure, and the brain, the development was extremely slow. And we found that, this is not, in this case, this is the karyotype, and the various aneuploidy is seen, and this phenomenon occurs during the culture. And this is the metaphase picture, and all chromosomes are separated while maintaining their bowed shape, and some are aligned with each other. And this is a very rare autosomal disorder, PCS-MVS syndrome. And this syndrome is associated with the BAB1B gene variant. And the BAB1G variant, PCS-MVS syndrome, is the very prone to child cancer, and with very, very poor prognosis. And this case was found that compound heterozygous inheritance of the BAB1B variant. And the BAB1B is a cell cycle checkpoint kinase, and it acts as a brake, preventing the cell with abnormal DNA or chromosomes from the proceeding with division. So the BAB1 gene, the gene causes checkpoint loss, the induced chromosomal instability, then may cause cell death. In other words, apoptosis over activation causes microcephaly. This is the last slide. Finally, we have to take into account that it is not merely a matter of size, whether the head is large or small, but